So I'm gonna talk today about spatial biology and specifically about spatial transcriptomics. And I'm gonna focus on this study which we published recently. That is focused on understanding what we can do with one of the platforms that got released recently. But to sort of start understanding this, I'm gonna go back in history and start explaining a bit what my PhD is. First of all, I mean, I'm gonna be talking about publicly different technologies that are commercially available, and I just want to make some disclosures. I'm co-founder of Spatialist, which is focused on spatialomics analysis, and Mats Nilsson, who is my PhD supervisor. He was used to be an advisor for the next Xenomics, but that was prior to the work that I'm presenting today. Right. So during my PhD, I focus on during my time in Mass Instant Lab, I focus on working with this technology that's called in situ sequencing. That is a technology that was developed inside the lab to profile tissues in a molecular level and retrieve the spatial location of transcripts within the tissues. And spatial transcriptomics, it's an image-based technology. Sorry, in situ sequencing is an image-based technology, which means that we employ imaging to decipher the location of individual transcripts inside the cells and in the context of the tissues. And it's a targeted method, meaning that we are actually able to profile a limited set of genes, but with a very high resolution. This technology is the predecessor of Xenium, which I'll be talking today, and that's why I thought it was worth mentioning. And I'm going to just highlight how this technology works because it's actually the molecular basis for this platform I'll be explaining things about. So in situ sequencing is focused on the design, or the first step for in situ sequencing is the design of padlock probes, which are these oligonucleotides that consist of, they are semicircular, and they consist of two arms that will be specifically designed to hybridize unique sequences in the individual transcripts that we pre-select. And it's composed by these two arms that will hybridize on top of the transcripts. And then once we have applied these different padlock probes to the tissue, we will create like a circular structure that resembles like a circle, and we will close the nick using a ligase that will allow us to create a circle in the tissue that then we will amplify using this molecular method that's called rolling circle amplification. And then basically we will be able to generate these molecules in the tissue that we will be able to visualize using iterative cycles of hybridization of fluorescent probes, imaging, and stripping. And this process, it was actually this method was very popular during the last 5 years. With several iterations of the technology, and it was very successful in creating the first molecular atlases of the tissues. But of course, it's a highly manual process because you need to go to the microscope every time you want to do a cycle, and it's really error prone, and it also involves several computational steps that are not that easy to handle, such as image registration or decoding. So it actually presents some difficulties. Fortunately, the startup that came out from the lab got acquired by the next Xenomics, and they managed to basically, based on this technology, create the Xenium platform, which is currently a machine, which is what you can actually see in this photo. And this was commercialized in December 2022, and in January, the Nilsson lab acquired 2 Xenium instruments, and then you can see them in the photo. Here, they are quite large instruments, but these devices enable you to actually generate molecular maps of tissues with subcellular resolution and for a selected set of genes. This is basically an improvement of the in situ sequencing technology. And at that point, we acquired the machines, and of course, we were experiencing the lab in generating this lab in these data sets and analyzing them. So we had a very easy question that is what my studies focuses on, which is what can we actually do with Xenium? I mean, how useful is this technology? What is the new biology we can learn from this type of data? And we sort of subdivided this into three like more specific questions, which is how does the data look like? I mean, how should we understand this data? How should we analyze it? How does it compare to other platforms? Because this is not the only technology that exists. There are similar solutions, and we wanted to see how good or how bad it performs. And also, how should we process and analyze data to make the most out of it without inducing artifacts or any potential errors that we have. And that's what I'm going to be talking during my seminar. And basically, to answer this question, we tried to assemble a large data collection. At that time, it was, of course, the largest, but now it clearly got, I mean, we have generated way more data sets, and now it's sort of small, but it contains 25 different tissues profiled from, I mean, different tissues. Those are tissue slides that were coming from different organisms, mainly mouse and human, and also coming from different tissues and disease states. So we had both healthy and diseased tissue from different organs. Just to basically give an overview of how the technology looks across different types of tissue. Also, we included both FFPE and fresh frozen in the study to be a bit more general. And we try to basically perform different tasks from preprocessing, method comparison, and colocalization, and many other ones on this set of datasets. Basically, the summary of this the datasets, you can see it here. Essentially, we have most of the datasets, they are from human, different types of cancer, mostly breast cancer. And as you can see from fourth column in here, most of the datasets presented around 400 genes, 300 to 400 genes. We also created different descriptors, but of course, this is still very general, and it doesn't give us more information than just basically very general statistics. So, very first question, how does the data look like? Basically, the Xenium data set, it's a pretty simple type of data. We can, here you have the visualization of the data. Basically, we have a point cloud of spots that present X, Y, and Z positions and the gene identity. So basically, we retrieve the subcellular location of every individual transcript. And we do it on X and Y axis, as you see in this picture, but we also retrieve the Z position. And this is something that is sometimes overlooked, because actually not all platforms do it, but Xenium can do it. And still we're processing tissue slides, which actually mean that the Z resolution is not going to be very wide. We might have like around 5 microns, but still 5 microns might give us important information, both on the biological side and considering that might avoid creating artifacts. So the first thing that we did was to explore the Z dimension, see if we can actually include this axis in our analysis, or if we should completely overlook it. And for this, we contacted Navity's Lab and developed together with them this tool that's called Overlaply, that basically tries to explore the Z dimension of these datasets. And for this, what this tool does is basically takes the slide and divides the molecular composition of the slide into, let's say, smaller slides that are the top slide and the bottom slide, and creates some gene expression embeddings, and then using cosine similarity tries to see whether on the Z-axis across the X and Y space we have vertical signal integrity or not, meaning whether we have similar expression on the upper part and the lower part or not. And the lack of this vertical signal integrity would mean that we might have different structures on the upper part and on the bottom part of the slides. And then we applied this to different data sets. Here I'm showing a small ROI where you can see an example. We managed to identify cells that were overlapping. They were, of course, not the great majority. We might have around, I think it's around 5% of the cells, or even less, but it's a significant proportion of the cells that are overlapping. And here you can see an example of an ROI that clearly presents a different molecular profile if we see it from the bottom or if we see it from the top. So summary of this is that Xenium contains that information, which is clearly overlooked in most of the analytical tools. And we should include that into our analysis if we want to perform a very robust analysis. Another interesting aspect that we also explore in our data is that is the subcellular resolution of transcripts. And basically, one thing that we realized by looking at the expression maps that we were finding in the tissues is that different markers that were supposed to be expressed by the same cell types presented a different spatial localization. And here you can see actually some of the genes, opaline and GFAP, but which are the most nuclear enriched gene in the case of opaline, and the least nuclear enriched gene in the case of GFAP. And you can clearly see that they have different subcellular distribution. And we even showed that one can use the location of individual transcripts on the subcellular level to identify subcellular structures. And that's something that some packages enable you to do now in a more robust manner. But we showed this resolution, which is sometimes overlooked in the analysis, because people try to segment the cells, meaning basically define the borders of the cells and just ignore all the other information that is present in the data, which can be useful in some cases. All right. So this is a bit how the Zoom data looks like, but of course we were not happy with only getting some statistics of this technology. We also wanted to compare to other available platforms. And for this reason, we designed a strategy to compare the Xenium data to other available technologies. And our strategy consisted of the following steps. We compiled a different set of data sets that were provided with different technologies, mostly image-based spatial transcriptomics, meaning that employ imaging and therefore have subcellular resolution and based on panels, so the same characteristics I mentioned before. But we also include Visium because it's a broadly used technology and the comparison was worth. And of course, for this I didn't mention, we use mouse brain. And the reason why we use mouse brain is that it's the type of tissue that, at that time, we didn't have that many data sets for different technologies, and all technologies presented some data sets in mouse brain. So it was the easiest to compare. And since we want to ensure a fair comparison, we ensure that only the same regions from the different data sets were included in the analysis. And for this, we perform regional annotation of the same structures across the different technologies, as you can see in the drawing here. And we redo segmentation, which is the process of defining the cell boundary. And the reason why we redid the segmentation was that segmentation can affect the comparison, and different technologies present different types of segmentation. Some of them are membrane stain based, some of them are nuclei based, some of them employ hybrid strategies. And in our case, we employ CellPost, which is a nuclei-based segmentation that's based on deep learning, very popular strategy. And we know it's not the optimal one, but we wanted to have like a reference, like common segmentation across different regions. And then we employ different metrics to compare the different technologies. So these are the main statistics for the different platforms. And basically, we have three main ones: transcripts per cell, genes per cell, and the number of profile genes. And if we look at transcripts per cell, you can clearly see that Cosmix, which is a Bruker technology, has way more transcripts than Xenium, but all the other technologies, they present a way lower number of transcripts per cell and also genes per cell. However, in this case, this has this actually a bit misleading statistic, because Cosmix at that point also managed to profile a larger amount of genes, which essentially means that they were actually getting more information per cell, but also they were profiling more genes, which really doesn't really tell us that much about the gene efficiency, the detection efficiency. So was the next thing that we did. We tested the detection efficiency of every technology. And for this, what we did is to compare the technology, and we compare the detection efficiency of every gene in every individual technology to the detection efficiency of a reference single cell dataset. And in this case, we use the Allen-Braille Cell Atlas, the ABC Atlas, that contains single cell dataset that we could match with the section that we use for this study. And then basically for every individual gene that we profiled in the different datasets, we compared its efficiency with the efficiency of single cell. Basically, 1 in here means that we have the same efficiency in single cell and in the spatial technology. And values above 1 means that we have a higher efficiency or higher detection efficiency in the spatial technologies. And as we can see here, different technologies present a pretty similar detection efficiency, but we could see that MC, which stands for molecular cartography, and Xenium are the ones that present a higher detection efficiency. However, most of them presented a pretty similar level, except for, in this case, cosmics that present a considerable amount of genes below 1, meaning that present a lower detection efficiency compared to single cell. But the number of transcripts we detect per cell is not the only metric we can look at. We also want them to be precise. We want them to be specific. And for this, we also put together a metric that we call NCP that measures the gene specificity for every individual gene. And basically, the NCP is it's a metric that essentially tries to quantify the lack of expression of so basically, if we take the expression profile of every individual cell type that we have from the single cell data, and we try to identify the genes that are not supposed to be expressed in those specific cell types, we can use the NCP score to quantify how many transcripts that were not supposed to be expressed in the cells of interest are actually expressed in those cells in the spatial methods. So this purity score basically the lower the worse, and allows us to quantify how good or how bad the specificity is. So essentially, with this we can try to we detect that Cosmics again is the technology that presents a lower specificity, while Xenium presents a slightly higher one, but it's not the one with a higher specificity. We have other technologies that, of course, have a better value. So basically, as the take-home message from this part is that different technologies might present different different characteristics, and then depending on the question you might have, it might be wiser to choose one or the other. But in comparison to, I mean, myself, that I've been working with this type of technologies for several years, what I really seen is that the different commercial products are sort of synergizing in their characteristics. So they are getting to a sweet spot, compromising between the number of profile genes, the number of transcripts detected per gene, and also the specificity. So they are starting to look more similar to each other than they used to do before. Okay, so we now understand how Xenium compares to other technologies, but still we don't know how to work with this data. So we try to, in the next part of the paper, we try to study or to explore different ways of processing the data. And I'm just going to go in the next minutes through different steps that we found to be crucial for processing this type of data. And the first, and maybe the most important one that I would like to emphasize quite a lot, is segmentation. And segmentation is basically the process of defining the cell boundaries of every individual cell. And in Xenium, at least originally, because now they have a multimodal staining that actually includes membrane stainings and so on. Originally, this was done based on DAPI. And here you can find DAPI staining. In white, basically, we can visualize the cell nuclei. And on top of those, we can, of course, visualize the transcripts, as you can see in here. Luckily, the Xenium, let's say, default analysis pipeline performs segmentation. And that's what I'm visualizing here. And the way that this segmentation works is that we basically segment the nuclei using a modified cell pose algorithm that they call. And then the next step that they do, typically after this, is to expand the cells. So you can see here a lot of the spots that are in blue, they would not be really assigned to any cell. So we would be losing a lot of information. So by sort of expanding the masks, we can achieve a higher number of transcripts assigned to cell. And now with expansion, what we call expansion, we have now assigned a larger number of transcripts to our cells of interest. So by doing that, what we get is expression profiles of every individual cell, and we can treat that as expression vectors from single cell data. So we can use the standard single cell packages, packages designed to process single cell data, such as CAMPI, CEBRAT, or many others, to basically define a population. So that's what I'm showing here. In a mouse brain section, one can see that it's pretty easy to identify different populations. It's very obvious in this UMAP, and then we can place them back in space and sort of generate molecular maps of these tissues. So I told you that segmentation was pretty a tricky process and quite something to really explore. And here I'm showing the reason why this is. So as I told you, the next segmentation is based on nuclei, and then they sort of expand the nuclei. And one thing that we did was to basically take these two expression vectors and try to cluster them independently. The one without expansion and the one with expansion. So only nuclei or nuclei plus expansion. And what we can see here is the same population, in this case oligodendrocytes that represent in the brain, labeled by area, as you can see on the plot on the right. Oh, sorry, on the left side. And one thing you can see is that whereas in the nuclei-based UMAP, all the oligodendrocytes, they fall in the same area, and they are pretty much merged. They don't really split by area. When we include expression that's based on expanded matrices or the expanded segmentation masks, they sort of divide by area. And this division by area is something that we identified, and now the next genomics people, they have corrected, I think, in their default software. But it's something that can be very, very dangerous because we would be identifying different subpopulations that in reality they don't present a different molecular profile, but they simply divide because the transcripts that are outside the segmented cells in every specific region, they are region specific. So we really want to avoid this, and we want to find a good segmentation strategy that maximizes the number of assigned transcripts while keeping avoiding these errors. And to explore this idea of expansion, we try to define first what the optimal expansion of the cells should be, right? We know that the default nuclei-based segmentation in 10x, it's solely based on the nuclei, which misses a lot of spots. But if we expand to the level that they were expanding originally, which was quite a lot, we are creating artifacts. So maybe there is a sweet spot in the middle between not expanding at all and expanding too much. And that's why we did this analysis. So basically, in this analysis, we assign every transcript to their closest cell, and we basically create the expression profile of every individual cell based on the distance to the nuclei. Then we basically define the nuclei expression as the ground truth, so the expression that the cell should have. And we know that there are some caveats to this, such as the local organization, subcellular organization of transcripts, but it was a good enough starting point. And basically, for every molecular profile at certain distances, we computed the correlation to both the nuclei expression signature and the background, which you have in blue and in orange, respectively, in here in the bottom left plot. And as you can see, at short distances, the correlation to the nuclei is very high. And as we move far away from the nuclei, it starts decreasing, and the correlation to the background starts increasing, meaning that we're actually not capturing relevant information anymore, but instead including more or less random information that might be coming from other cells or so. So we did this for all the different cells, which is what you can see on the right plot. And we realized that different cell types present different optimal expansions. And that makes a lot of sense, right? Biologically speaking, different cells will have different sizes, meaning that a rigid expansion of the cell boundaries is not something we can rely on. So so it sort of makes sense to expand a bit the cells, but after certain ranges, such as in this case, distance of 5 microns, it starts to be a bit meaningless because different cells will have different sizes and so on. I mean, this analysis was very clear to us, and we clearly needed a better method for segmentation. So the next thing we did was to try different available tools for segmentation. And the challenging part in here was to define what a good segmentation is, because it's not so obvious, right? We want to define well the cell borders, but we want to quantify that. So the way that we quantified that was using this sort of two axes, these two main metrics. First is the proportions of reads assigned. Of course, we want our algorithm, our segmentation algorithm, to maximize the number of transcripts we assign to cells. right? Because we assume that most of the transcripts or all the transcripts that we have in the tissue, they should come from some cell. So we want to maximize that. But at the same time, we want to also not oversegment, not induce errors, right? So to quantify this, we put together this negative marker purity, which is a metric for specificity. And essentially what we took a look at is the lack of co-expression of genes that were not supposed to be co-expressed in a reference single cell dataset. An example of this is if we have two one oligodendrocyte and an astrocyte marker in this case, or a fibroblast and an endothelial marker, that we know, because we have a reference single cell, that they should never go together. We sort of evaluate how many times they co-localize in our tissue, in our segmented cells. So essentially, with this metric, we managed to quantify how well the segmentation works. And of course, the larger in this case, the more specific it is, the better it works. And of course, these two metrics are tightly linked. I mean, the more transcripts we assign, the more likely it is to induce segmentation errors and therefore to lose specificity. And that's what you can see with most of the algorithms you can see in here. The different shapes represent different algorithms. And I will also include the Xenium default nuclear segmentation and cellular segmentation, which is what I showed you before, the unexpanded and expanded segmentation, which are shown with the stars, or sorry, the crosses. So basically, we could really say that no nuclei-based segmentation method outperforms the Xenium segmentation. But we did find a method that was outperforming it, which is this method that's called Baser. So Baser is a segmentation method that doesn't only rely on the nuclei position or the nuclei staining, but also includes the RNA local composition of the cells, so the location of the spots and their density, among other things. In a Bayesian manner, it sort of assigns in a non-rigid way transcripts to individual cells. And these are the points that you see here in blue, with different prior segmentation confidences, because this algorithm relies or can partially rely in prior segmentation that's based on nuclei. And one thing that's very obvious is that all these blue spots, they are above the diagonal that we have for all the nuclei-based methods. And this sort of agrees with or matches quite well with what I showed previously of the different optimal segmentation for different cell types. Because these type of methods, in this case, we included only Baser, but we might have other methods in this category, such as PCI-SEG. It's called Seger or ComSeg. There are several segmentation methods in this group that are basically non-rigid and rely on the location of transcripts. They cannot outperform the other ones because they are a bit more flexible on the way of assigning transcripts to cells. So, but another obvious take-home message from here is that no, we don't have a perfect method for segmentation. All methods either do not assign all transcripts to cells, or they lose a bit of specificity and can be prone to errors. So a take-home message from here would be that segmentation is not really a solved issue in spatial omics, and we are not really sure if it will be. It's a solvable problem with the tools we have currently. Okay, so another thing we did after segmentation, as I told you, we can employ single cell or tools developed for single cell to process spatial data. And the steps for processing the spatial data are not non-trivial as well. I mean, people are sort of using the same steps that were developed for single cells, such as normalization, log transformation, etc. But it is not obvious what's the best combination for that. So we basically, to assess that, we tried to put together a workflow that was based on simulating spatial data based on a reference single cell data. So basically, we were taking single cell data from cell by gene and turning it into a Xenium-like data set. So basically, we were reducing the number of genes profiled. We were reducing the detection efficiency. We were transforming the gene counts by also inducing some segmentation errors and so on. And then we were trying different strategies to process the data. And with that, on a very empirical manner, we try to identify the best way of processing the genome datasets, which ended up being actually pretty similar to what is done in single cell, but we wanted to test it empirically. So from our study, out of the combinations we included, the best way of processing the spatial data sets is to actually perform a library size-based normalization. Recent preprints have shown that there are some caveats in here and that this can induce some errors for downstream tasks such as differential expression. But out of the combinations we had was the best. Then log transformation. It is important or it is it's essential not to select for highly variable features in here. And the reason why this is makes a lot of sense, we have a limited number of genes. We have 300 genes in these panels, which means that we would be losing a lot of information. I mean, most of the genes that we have in these panels are already pretty selected to be useful, so it makes a lot of sense. And then scaling seems to be useful also for identifying the cell types correctly in this case. So the overall strategy we find to be the best one was pretty much the same that is used in single cell with some small differences. But we find these workflows to be very good in retrieving the main, or we find the steps to be useful to retrieve the main cell types present in the dataset. Another task that we explored was the selection of especially variable features. So in an analogous manner to the highly variable selection in single cell, the spatially variable features identification consists of identifying the genes that vary their expression across the space. So basically that in a part of the tissue, they present a high expression, and in another part of the tissue, they present a low expression. And we benchmark the performance of different tools. And here we have the list of the tools from ScanPy tools to SquidPy tools to Symfony and other tools. And of course, we don't really have a ground truth in here. We don't know what a spatially variable gene is. Because I mean, there is not like a very hard cutoff between what does it mean to be variable or what. We don't really have a good definition, but still we could assess we could compare the performance of the different tools. And one thing that we identified is that all available tools or all available tools included in the study, they present a very similar performance. And this you can see in here, basically on the plot on the bottom right side, you can see the different methods with every gene rank from the most especially variable to the least especially variable, and you can see how for all the methods the matrix turns darker as we move to the right, meaning that there is a high consistency in the performance of different methods. And then different methods we identify to present different ways of assessing statistical significance, meaning that different methods presented a larger or lower number of spatially variable genes detected based on how the statistics were performed or which test was used. So overall, all methods look pretty much similar, but there is a big difference in the way that the final spatially variable genes are identified.
Hey, so there's one question in the Q&A session.
Yep.
Do you want to save that question at the end, or you want to answer that right now?
Yes, I can answer now, but I don't really see the chat. So if you can just.
Oh, okay. Yeah, I can read that. So I think this is the Baser, right? The one of the tool you have used. Can Baser be applied to correct the original Xenium cell advanced segmentation? Is it better to start from nuclei segmentation or full cell segmentation?
In our study, we tested Baser with like prior nuclei segmentation and without, and it was working better when it had a prior nuclear segmentation. And the reason for this is that the nuclear segmentation is actually pretty precise, especially in the datasets where we applied it to, where it's not that tricky to segment the nuclei. And it was actually adding some additional information that Baser per se couldn't really do it. However, there is a caveat here, which is that we didn't really test it using multimodal segmentation, meaning cellular membrane-based segmentation. And the reason why we didn't test it is that it wasn't really released when we started with the study. So in case you have both nuclei and membrane-based, I'm not sure what I would choose, but I would add some prior to Basel. I think that that's something that seems to be helping, at least in the definition of individual cells. Because the problem that Basel might have, and that's something that's reported in the original manuscript and so on, is that it fails to identify individual cells in very homogeneous regions. Meaning that if you have, for instance, an epithelium and you have a lot of epithelial cells next to each other and they have a similar expression profile, the algorithm will fail to distinguish one from another if they are very homogeneous. So in that sense, the nuclear segmentation prior can help distinguishing these individual cells.
Okay, there's another question from the audience. I think Su Guangyu. Sorry, I'm not sure I pronounced it correctly or not, but I think you can unmute yourself and then ask a question. Can you unmute yourself? All right, I think maybe well, he has problem to unmute himself. Okay, so you can basically just right click your name and then just say unmute. Maybe we can save the question. It seems like he has technical issues to do that. Sorry.
Yep.
Oh, same question. All right. So he said that he has same question. All right. So you will answer that. All right. So you can go ahead, Sergio. Basically, the same
Yep.
question as
Okay.
the
Yeah.
paper.
Yeah. Sorry.
Yeah.
Okay. Good. So yeah. So we also evaluated a couple of more. I mean, tools applied to Xenium for a couple more tasks. First is gene imputation. So gene imputation, I mean, as I mentioned at the very beginning, we don't really profile the entire transcriptome currently or with Xenium. In these datasets, we were limited to 300 genes. Current datasets are limited to 5,000 genes. So it increased the panel size, but still we're limited. And basically this task consists in using a single cell dataset of reference, trying to impute the expression of the genes that are not originally profiled. And for this, there are different algorithms. And we benefit from a recently published benchmark comparing these algorithms, and we sort of reuse this benchmark from Lee et al. And essentially, we try to see how the performance of different algorithms was looking, and we identified some of them outperforming others. And especially, we were especially happy with the performance of speech, and the Seurat one was also looking pretty nice. So we could define best practices in this case. But maybe another even more interesting part in here is that the performance of the imputation, we can assess it on a per gene basis, meaning that we can assess whether we can impute a certain gene well or not. And one thing that we found is that for individual genes, the performance across methods was pretty much consistent, meaning that for some genes we can impute very well their expression profile, and for some others we cannot impute very well the expression profile, or we cannot impute at all, resulting in an overall limited performance of these methods, which is something that we can see when we also look at the overall statistics. So for some genes we're very good, for some others we are not that good, and we were actually wondering why is that the case. So we tried to explore different metrics, and we showed some of them to be correlated with imputation accuracy. We saw that the mean expression of the gene was somehow correlated with the imputation accuracy, which makes sense. Noisier genes will be less likely to be correctly imputed. But most importantly, and with a Pearson correlation of 0.7, we found a correlation between the imputation accuracy and the mean correlation with other genes present in the panel of a special transcriptomic profile. And this is very meaningful because it actually means that these algorithms, they work very well with genes that present a very similar gene in the panel that we profile. So basically, they are very good in sort of making imputation of for instance, cell type marker genes or these type of things. But with more complex profiles, it's way more difficult to do it. And essentially, if we have genes that do not follow the expression profile of any of the other genes that are present in the dataset of interest, it will be very difficult or almost impossible to retrieve their expression profile using imputation algorithms. So the last task we evaluated was domain identification. And in here, domain identification essentially consists in the identification of cellular structure or supracellular structures that we might have in the tissue. And we don't really have a ground truth per se, but we do have in the mouse brain very good references and some atlases with different structures. So we could annotate the different structures that we might have in our datasets, and we assess the performance again of different methods. And here I want to add a caveat, which is that we only use the expression of the cells for the domain identification, and some of these methods, they also have the option of including histology, which is something that we didn't really include in this story, but can be useful to identify the boundaries of structures and to better define the borders. And essentially using these, we could see how this sort of binning-based domain identification and as well as Banksy were the top performing algorithms that sort of retrieve better the original manually notated regions based on the atlases. Yes, so as a summary from our study, we saw that Xenium, or we confirmed that Xenium is one of the ISD platforms with the highest detection efficiencies, and it also presents a pretty high specificity. Datasets contain useful, of course, cellular and tissue information, but also subcellular and some three-dimensional information. Segmentation is certainly key in Xenium data analysis, and this applies to other spatial omics technologies. That is the biggest challenge on the field. And of course, selecting the correct algorithm can maximize the results, can enhance the performance of the output of our analysis, and it is important to really see which tools work better with which datasets. With that, I would just like to thank everybody who was involved in this study, especially Mats, who's my PhD supervisor, but also Navid developing the overlapping tool, Carolina Belvi working on Uppsala University on the subcellular part, and Fabian Thijs, who is my current postdoc supervisor, as well as everybody in the team. And I'll be happy to take questions. Thanks.
Well, thank you so much, Sergio, for the seminar. And now it's open to questions. So I see one question from the chat from Ashish. So did you also explore the spot annotation tools for CCM data?
I'm a bit confused about the term spot annotation.
Hey, hi. So basically, I'm like asking whether the cell kind of annotation tools that you use for this data set, and how do you look at those? So yeah, thank you.
So we didn't explore the annotation tools in this study, unfortunately.
Okay, thank you.
So I have a question. I guess it's more for a open discussion. So I was curious about what your thoughts are with regard to kind of the sensitivities for performing differential gene expression analysis with this type of in situ based approach. I mean, we know that with VCM or even VCM HD, that is very feasible. But that's a NGS-based approach. But with this in situ and imaging-based, and I'm not quite sure, because there's going to involve some sort of hybridization, signal amplification. I know it's almost like a single molecule resolution, but how sensitive it is to pick up the subtle gene expression differences.
I think it's pretty. I think sensitivity in here is not really a problem. I agree that the Visium and Visium HD might have this problem, but that's not the case. There are two things to consider, though. The first is, I mean, the inclusion of the genes of interest in the panel. Of course, if you don't really have those genes, you cannot really assess them. And the second one is segmentation. If segmentation can affect quite a lot of differential gene expression, especially if you don't define well the cell boundaries, it is very likely that you might induce some errors. But from the sensitivity point of view, we do have typically a higher sensitivity than compared to single cells. So we should be able to make even statistically stronger claims regarding differential gene expression. Also, because something I didn't mention is that the number of cells we typically profile in these datasets it can be an order of magnitude larger than single cell. So it's a very suitable method for differential gene expression analysis.
That's great. See, oh, there's Benjamin. I don't know if this is from panelist or I can read the questions if not. Okay, so the question is the nuclear versus expanded comparison was striking with respect to differentiating oligodendrocytes. Is there data on single nuke RNA sequencing versus single cell RNA sequencing demonstrating the same thing, or is this for some understood reasons and in situ technology issue?
Yep. So we haven't really checked single nuke versus single cell because we think that these differences we see are artifacts from cementation. So that was the easiest way of justifying that, or not the easiest way, but the most. I mean, we showed that with the subsequent analysis that differences we were seeing between regions are actually deriving from the composition, the cellular composition of the region where the oligo was, which means that we are not really seeing, in that case, changes between oligos and nuclei expression and cellular one, but instead we are sort of inducing these errors. So we don't think there is difference between cellular expression, or we cannot really say that there is difference between cellular expression in nuclei and cell. It's more like an error that we see in the datasets when you don't really segment well.
So I had another question. So we know that some other platforms like I think might also be 10x offers platform that allows simultaneous RNA profiling and a protein marker profiling, right? So in that case, wouldn't that be advantageous for kind of to help to delineate the cellular boundaries for segmentation? I mean, I know this is very different technology. Yours is in situ based, but would that even be a possibility?
Indeed, yeah, that's a possibility, and that's something they are. I mean, now they have their kit that's commercialized together with this technology that enables you to stay in the cell boundaries. Regarding the cell boundary staining, I think there are a couple of comments I wanted to add. The first is that as nuclei, for instance, it's a pretty universal, DAP is a pretty universal nuclei marker, and it stains all cells. Right now, 10x is claiming that their membrane staining kits and so on stains all the cells, or a good chunk of the cells. And this might be pretty tissue dependent and cell type dependent because it's based on antibodies, and some proteins might not be present in certain structures in different cells. And the second thing is that even with those, the performance of the cell segmentation is not optimal. We still see a lot of dense aggregations of transcripts outside the cells that are not nicely segmented with these stainings. So those might be they are an improvement in comparison with only nuclei segmentation, but they do not really solve the problem entirely.
I see.
Yeah, there's one more question in the Q&A session. Do you want to read that question again? I think that's from Eugene again. Well, I can read that. Okay.
Oh, sure.
Yeah, so that's one more question. So from Eugene, I think he actually asked the first question was, so in your experiences, how long of the FFPE blocks at +4 are compatible with Xenium's pipeline? Sometimes it can be difficult to have sample collected at the same time. Is a couple of months acceptable?
So I'm not myself. I know that FFP is compatible, and I know that it is typically performing a bit worse than phase frozen in Xenium, but also in other technologies. I'm not completely sure about the times. I cannot really say much because I'm mostly focused on the computational part. So I unfortunately cannot really say much.
I guess we just only have a few minutes left here. I was wondering if you can provide us with some final insights about how to make decisions to choose between in situ imaging-based spatial transcriptomics versus NGS-based spatial transcriptomics. Like in what kind of study design, which one would be more beneficial than the other?
Yeah, I really think it depends on both your research question and your tissue. I think the overall advantages of each of one is you would go for image based if you do care about the subcellular or the cellular resolution. You want the profile of every individual every individual cell, and you care about profiling individual cells in the space. And you want a high detection efficiency. So you want a lot of transcripts per gene. And then if you would go to NGS based, if you really want to do an exploratory analysis, you don't really know, you just want to understand the new biological system and disease or something, and you don't really have any prior on that. You just want to. So yeah. But I have to say that the bridge between the technologies is closing. The biggest disadvantage of image-based was the limited panel size.
Right.
And for instance, now CosMX from Bruker has like a whole transcriptome profiling. Xenium has 5,000 genes, and eventually I guess they will go to whole transcriptome as well. And Visium HD on the other side, for instance, or Visium HD, but also StereoSeq and many others, they are making I mean, Illumina, for instance, with the recent product, they are making the resolution smaller and smaller, so that they are actually, both types of technology are getting more similar.
Great, thank you for your insight. I guess if there's, go ahead.
No, I think that's great. Well, I think seems like we don't have more questions from the audience, so maybe we can stop here. Well, thank you guys so much, and then a very good, interesting presentation also.
Thanks so much for hosting me. And yep, if anybody has doubts, I'll be happy to take questions also on email or something.